Thank you so much for the honor of being here on what a distinguished program. I want to thank Richard especially. I was talking to a leader in environmental protection. I'll just say from the West Coast, and she said, "Do you know Richard Dennison?" I said, "Yeah, he's a really good human being." And she said, "Oh, he's a national treasure." All of a sudden, a good human being didn't sound so good. I thought I was being nice, but you're a national treasure, evidently. And so, I also want to thank Chris and Bill Farland. Bill, his distinguished career at EPA, left a foundation of great science, and it's a pleasure to be following up. And I know you're doing great things where you are. I also want to thank where's Hal? There's Hal, who leads our health effects lab down in RTP. I have the pleasure of working with him as he does great things down there. I'm going to I get the opportunity to be sort of the overview guy. So there's people here in this audience that know just about everything there is to know about green chemistry, and others who know a tremendous amount about toxicology, and maybe some that know a little bit about both. But in order to try to bring folks to the same level, I'm going to give a bit of an overview. But before I hop into that, I just want to say this idea of bringing together the toxicology community and the green chemistry community, having problems meet solutions, is something that has been recognized as tremendously important, tremendously essential. And I want to call out two people who have led efforts, Pete Myers and Karen O'Brien, the leaders of Environmental Health Sciences and Advancing Green Chemistry, respectively, for their work and doing this over quite a while now, bringing these communities together. So I want to thank you. Now, I'm not going to get down into the weeds as much as I like the weeds. I really enjoy the weeds. The weeds are good places. But Dave Dix is going to be talking a little bit about some of our efforts at EPA, and Annalena Vuchkova is going to be talking a little bit later this afternoon about some of the work that's going on at Yale University. So, well, I might mention in passing, those are some talks. Well, all of the talks, the ones that you want to really follow. Last thing I'll say before hopping into it is when you look at green chemistry, it's a proven, demonstrated field where it can bring about environmental improvement, benefiting human health, benefiting economics and profitability at the same time. The question is about scale. We have thousands of examples, thousands upon thousands of examples. The question is not whether or not it works, is how do we bring it to scale with the urgency that's necessary. And you want to know something? All the scientists in the room, I'm not worried about the thermodynamics. The thermodynamics are on our side. We're going to get there. What we need to worry about is the kinetics. Are we going to get there fast enough? Are we going to get there at the rate that we want to? And to take this metaphor just a little bit further, what we need is catalysis. And this workshop is an important catalyst for bringing about the transformation so that problems can meet solutions in an effective way. So a little bit of an overview on green chemistry, if I may. The simple one-sentence definition of green chemistry: the design of chemical products and processes that reduce or eliminate the use and generation of hazardous substances. Well, that's pretty straightforward. It's one sentence. Well, if one sentence is going to define a field that is now worldwide in scope across so many industry sectors, it really should mean something. And perhaps more than any other word, the word design is essential in this definition. It's about the thoughtful approach about how you make the molecules and the pathways by which you go about synthesizing these molecules so that they're not using and generating hazardous substances. Now, can you reduce waste by doing green chemistry? Absolutely. Can you make a process more efficient by doing green chemistry? Absolutely. Is that where it ends? And the short answer, of course, is no. It is all across the life cycle, from the beginnings of feedstocks all the way to the end of life and beyond, and we'll talk a little bit more about that. Now, when we're talking about the realized benefits, not hypothetical benefits, the realized benefits that have been demonstrated, it certainly goes across environment, human health, economics and competitiveness, sustainability, and new scientific knowledge. Now, for a few of these, they're directly relevant to today. The reason that, as much as I'd like to be here for the entirety that I need to leave, is because across town, the National Academies is launching something called a project on sustainability linkages. And this is showing how all of the different efforts in public health protection and environmental protection are interconnected across agencies and how to bring those together. I then fly down to North Carolina for a session on competitiveness and sustainability that Vice President Biden is scheduled to speak at this evening, and to a CEO forum. What we're seeing is these benefits of green chemistry are an essential and fundamental part of meeting these environmental, public health, and competitiveness goals all at the same time. Now, I often say that if I were to claim that green chemistry has the potential to affect everything from electronics to aerospace to cosmetics, agriculture to energy, that would be an awfully bold statement. But that's not what I'm saying. I'm saying that it already has. These are some of the industry sectors where it has already been incorporated with award-winning technology recognized by the Presidential Green Chemistry Challenge Awards. All of these award-winning technologies have been put in place because they're profitable. Not because there's a regulation that says thou shalt do green chemistry. There's many ways of achieving your environmental and health standards. But because people know that by pursuing green chemistry, they can meet these dual goals simultaneously. Now, I say that it is moving our consideration of our environmental issues from the circumstantial to the intrinsic. Well, what the heck does he mean by that? Well, what I mean by that is that rather than looking at the conditions, let's see, is this thing working? Can you hear me? All right, great. This will work much better. Rather than trying to simply change the circumstances by which we either use or transport or have protective gear, goggles, respirators, we start thinking about how can we change the inherent nature of the materials that we use. So we can put smokestack scrubbers on smokestacks. We can wear these gloves and goggles, and we know that all of these, as a probability function, can and will fail at some point. And when they fail, the risk goes to its maximum because you haven't addressed the intrinsic hazard side of the equation. When you deal with the intrinsic nature, molecular design for reduced toxicity, etc., you're able to impart new performance, new capabilities, new efficiencies at the same time as you're meeting these other goals. Now, the circumstantial approach always has to be a cost drain. It cannot add value, it cannot add performance, it cannot add capabilities to your product or process. So this is I was going to say dirty little secret, this is the clean little secret that people are catching on to throughout different industry sectors. Because our basic, in its most basic form, our risk calculation is hazard and exposure. Virtually every circumstantial approach is trying to lower exposure, takes the hazard almost as fixed. You have this chemical, therefore how do you stop your exposure to it? And in some cases that may be necessary. Green chemistry is dealing largely with the hazard side of the equation. But can you design a molecule or a manufacturing process through green chemistry that even reduces exposure? Certainly. And also, of course, green engineering approaches. Now, I don't put this up as I often say, an eye chart, but these are the 12 principles of green chemistry. And I put this up only to communicate that this is not something where it's a noble wish. We're going to do things that are good and clean and sustainable and leave it up to sort of an amorphous, non-defined, without a framework type of approach. This is saying there's ways of going about it. This is the how of sustainability, and this is what the chemistry of sustainability looks like. And if you look at those 12 principles, you'll recognize that it's not just across one step in the life cycle, perhaps a manufacturing step or whatever. But no, rather it's across the entirety of the life cycle. What are the nature of the feedstocks and the materials that go in? What's the nature of the energy? How do you ensure that you're not generating waste? That every atom that you put into a process winds up in your product, and on and on. So it's systematically looking across the entirety of the life cycle. What we're here to talk about mostly today is what is really embedded in Principle 4, as well as others, but primarily in Principle 4, which is the design of the molecules themselves in order to reduce intrinsic hazard. And how do we go about doing that? So, what kind of progress has green chemistry made over the past 20 years? Well, scientifically, there's quite a number of journals. These are 3 of the most well-known green chemistry letters and reviews. Green Chemistry by the Royal Society, Chem Chem. There's others, Green and Sustainable Chemistry, an online international journal. There's one point that I do want to make is that the journal entitled Green Chemistry by the Royal Society has a higher impact factor than the more than a century-old Journal of Organic Chemistry by the American Chemical Society. So when we start looking about rigorous science and how it's making an impact, that's something to note. Now, articles on green chemistry, various papers, and virtually not just, oh my goodness, my slides aren't quite working. I think we need more CPU power there. But there's quite an increase over time of the papers being published. Let's see if this one works. Not quite. There's an extraordinary amount of press coverage and popular demand for green chemistry. There's research networks around the world, of which we are showing about half of this slide. We have many of them represented here. In 33 countries around the world, there are research networks around green chemistry. In China alone, I just returned from China, I guess, about a month ago. They just recently launched their 13th key lab on green chemistry. Now, when we look at the science that green chemistry is advancing, I am old enough and have been around this field long enough to remember when people said, green chemistry, it's all about bio-based materials. Then it was green chemistry is all about aqueous solvents. Pardon the reformatting of the slides. It's all about aqueous solvents. And that green chemistry is all about just organic synthesis and synthetic methodologies. And that green chemistry is all about molecular self-assembly. And it's all about next generation catalysis and bio-based transformation and biochemistry and alternative energy and green nano. I see Jim Hutchison right in the back, the pioneer of green nano, or room temperature ionic liquids or degradable polymers or molecular design of biofuels. And of course the answer is yes, green chemistry is about all of those. Because it is working at the molecular level, it really doesn't know or care whether it's going to be in a particular product in a particular sector. It knows what its basic physical chemical properties are. It knows the transformations it's going to undertake, and is susceptible to design so as to reduce the hazards associated with it. So it shouldn't come as a shock that all of these areas that have been pioneering areas of science by green chemists around the world are the current legacy of green chemistry, as well as supercritical fluids. Any one of these areas is enough to transform an industry sector. Now, there are businesses around the world, more than we can ever list on a slide. But I think the point is this. If I were to try to name off all of the companies, the products that have engaged in and utilized and implemented green chemistry, I'd say, there's good news and then there's better news. The good news is that there are thousands of companies, thousands of products that have been reinvented using green chemistry. The better news is that represents a small sliver of the power and the potential of green chemistry. For every one product or process that's been reinvented, there is certainly 100 or more that haven't even been thought of in terms of green chemistry reinvention. So there's tremendous upside potential. There's tremendous work to be done, and I view that as a good thing rather than bad. Now, this is a report that came out of Pike Research. It's a market analyst report. Came out about, I guess it's a couple months ago now, and talks about green chemical industry to soar to $98.5 billion by the year 2020. So one of the things that we're seeing is that leaders in various sectors are recognizing the business opportunities for green chemistry, and sometimes it's for different motivations. And quite frankly, I'm not sure I always care about the motivations. I really do want people to become very, very wealthy by doing good things. I really do. And so whether it's because of efficiency, because of market reputation, because of altruism and caring about the next generation of people on the planet, I really just like that good things are happening. There's conferences around the world, some of which you see here. There's networks of environmental groups and NGOs that are coming together to advance green chemistry. And then we have some of the concerns that are out there. A lot of headlines about whether it's contaminants and everything from bibs and baby bottles or toys or bug repellents or playgrounds or, I was going to say name your poison. Whether you are of the perspective that this is just a perception problem, or whether you're on the end of the spectrum, that this is a tragic poisoning of our current and future population, wherever you are along that spectrum. This isn't good for anyone. This isn't good if we're looking at people's health, and you're concerned about people's health, you know that this is a tragedy. If you're looking at people's businesses, and perception of companies and profitability, it's not good from that perspective either. And anywhere along the continuum, everyone can agree that this is bad. So, what do we do? Well, design for inherency. Design for inherency, molecular design for reduced hazard, that you'll be hearing an awful lot more about at a much deeper level in a little bit, speaks to, again, not changing the circumstances of a molecule, looking at its inherent nature. One of my favorite quotes from Aristotle is, "The term intrinsic nature does not indicate a factor's temporal status, but rather refers to its underlying and defining nature." And let me just take a moment to state the obvious. A molecule can do what it does in its performance, whether its performance is to be red, whether its performance is to be brittle or elastic, whether its performance is to conduct electricity, whatever its performance is, it does it because of its physical chemical properties. That's why a chemical does whatever it does. And do we know how synthetic chemicals get their physical chemical properties? The molecular architects give it to them. They give it to them, ideally, by design, through thoughtful design. In the same way that a molecule has sometimes certain commercial properties that make it of value, those exact same physical chemical properties are the ones that impart its hazards, its toxicity, its concerns, its ability to bioaccumulate, its ability to cross biological membranes, its ability to bind to receptors. And so it is within the power of the molecular architects to design properties of hazard and toxicity in the exact same way they've always designed the properties for function. These are a few references, and I rather than these references, I think the best thing that you can do is listen to Dr. Vuchkova, because this will point you in the right direction. But when we're talking about hazard, today we're going to be talking mostly about toxicological. But in the same way that we have concerns for physical hazards, whether it's explosivity, corrosivity, flammability, are we able to design those properties to reduce those hazards? Absolutely. Global hazard, whether it's depletion or persistence and stratospheric ozone depletion, greenhouse gases, are we able to design for those hazards? Absolutely. Now, what we're talking about today is what are the design handles? What are the design handles that a chemist has in order to modify or invent a molecule so that it's less likely, less able, less intrinsically capable of manifesting hazard? And as we think about toxicokinetics and the spectrum to toxicodynamics, you'll see that we look at the basic fundamental properties related to things like absorption, distribution, metabolism, excretion, molecular weight, volatility, particle size. Are we able to modify those? Of course. Are we able to understand how solubility and electronic charge of a molecule is able to play a role in a molecule's ability to be hazardous? What about as we go up? Shape, molar volume, bioaccumulative persistence, reactive functional groups. Of course, all of those play into it. Of course, those are standard fare in a molecular designer's toolbox of how we modify molecules. It's certainly, as we go up, it gets increasingly complex in dealing with key receptor binding events. And how do we understand this complexity? So at the base of this pyramid, there are things that we understand very well and easily can modify. At the top of this pyramid, there's no doubt that there's tremendous complexity and things that are far more complex to begin to understand how to modify. I don't think anyone in this room who's not running a fever would say that we've got all of the science that we need today in order to do this perfectly. That would be, in my estimation, an irrational overstatement. What's so important is that we do understand that there are steps that we can take even today, and we'll hear about some of those approaches, where we can not only have the conceptual construct about how to go forward, but there's steps that can result in very important leaps forward in molecular design in order to reduce hazard. So, are we able to reduce the potential for undesirable absorption in the same way that for certain therapeutic drugs that we want to make sure that it's able to be absorbed? Do we want to turn that model on its head and be able to reduce the potential for absorption, limit distribution, enhance metabolism to non-toxic products, and facilitate excretion of xenobiotics? So yes, in the same way that we know how to bring to bear some of the molecules that are, in many cases, life-saving or increasing quality of life, we know how to turn that model on its head so that we can increase the probability of designing a safer molecule. This slide sadly didn't come up, but what are we striving toward as a community? What are we striving toward? And really, the goal is a set of design rules, so that when you're introducing a new substance into the universe, into the world that the world has never seen and quite frankly doesn't know how to deal with, we have a set of criteria where we begin to understand that we dramatically lower the probability of the substance being able to manifest hazard. Whether it's going to be a certain range of particle size or ionization potential or log P of molecular weight, et cetera, et cetera. And these are found in some of the papers that Dr. Vuchkova will refer to. Now, oh, that's a shame. One more, that was a slide that is describing whether or not, by understanding what combination of physical chemical properties are directly related to a molecule's ability to cause toxicity, to cause hazard, by knowing which of those physical chemical properties, which subset of those 2, 3, 4 properties are crucial, can we then in multidimensional space define an area of high probability for toxic substances, high probability for safer substances. And I think that later today you'll be hearing a lot about progress being made in this direction, that with far higher probability that we can make steps in this nascent journey. So what I'm going to do is just close with something of a question and ask, should we be rethinking what performance means? What performance of a chemical substance, of a chemical material means? Is it enough to introduce a new substance into the world, a new material, a new molecule, and have its definition of performance only be for its commercial function? Whether it's color, strength, adhesion, on and on, whatever that narrow function is. Or do we have to include in the definition of performance that it also is required that the substance does not cause harm, has a reduced ability to manifest hazard, carcinogenicity, endocrine disruption, neurotoxicity, And if we are able, even at the beginnings of being able to design out or to at least a reduced potential for a substance to manifest hazard, that we have the beginnings of the scientific understanding and tools to move in this direction, what is our obligation to do that? So I'll leave that as an open question. One last thing that I just want to say before I turn it back to Richard is that this area of molecular design, this area of green chemistry, is at the foundations of what we're all striving toward and moving to sustainability. Sustainability across our various systems. It's I do say that it's the chemistry of sustainability, and something that I just want to let people know about. A National Academy study that it just came out, was launched this past Thursday officially. This is referred to in the vernacular as the Green Book, Sustainability and the U.S. EPA. I have been on the receiving end of a lot of NAS reports in my day. It is my opinion that this is second to none in terms of its quality and thoughtfulness. For the folks that I work with and around, I say it's about as close to required reading as a report like this gets. And one of the things that is embedded in this very thoughtful document is the role of green chemistry and environmental health sciences and moving forward in a sustainable systems. So with that said, I just want to thank you very much for the opportunity to speak with you. I am tragically sorry that I'm not going to be able to spend the rest of the day with you today, but thanks for the opportunity to talk. Appreciate it.
Paul, can we keep you here for maybe one or two questions? I'm going to start with one. I was very intrigued by your idea of turning pharma on its head in some ways, to think about designing out some of the things that the pharmaceutical industry has been trying to design in. It struck me because many of the methodologies that are emerging for toxicity testing are also arising out of pharma and out of the research fundamentally that's going on in that area. Could you elaborate a little bit on that? And I realize it's not necessarily a perfect analogy, but it seems to me a nice way to think about conceptualizing what we're trying to get at here.
No, thank you. So one of the things that is so important is, I often say that, well, Newton said that he could see the horizon because he stands on the shoulders of giants. What was unsaid is that the giants couldn't see the horizon. And so people have built tremendous tools, have gained tremendous insight about molecular design, tremendous insight about the synthesis, manipulation of molecules in order to bring about biological effect. Now, those tools have largely been developed in the pharmaceutical industry. And taking that same understanding of how you want to design a molecule so that it stays stable on the shelf, when you ultimately take it, it's going to be able to get into your body, cross the necessary biological membranes, get to its receptor, cause a therapeutic effect, and by the way, when it's excreted, its metabolites are innocuous and go into the environment. No small challenge, right? What this molecular design for reduced hazard is all about is how do you make sure that molecule, when you take it, when you're exposed to it, when you inhale it, when you somehow receive a dose of it, is unable to either get into your body in the first place, unable to cross the biological membranes, unable to bind to a particular receptor so that it cannot cause a toxic effect and is removed innocuously from your body. The work that's being done on high-throughput screening that David Dix will be talking about later is gathering a treasure trove of information, not on just a chemical at a time or a few chemicals at a time, but hundreds of chemicals across hundreds of bioassays. The use of this information will be tremendously powerful in informing our understanding of the problems and the challenges we have. The place where this information becomes transformative is when it informs our design, gives us insight on not just what our challenges are for the current generation of chemicals, but how to design the next generation of chemicals so that they're unable to cause these kind of hazards.
Maybe one other question. We're going to have to move on, unfortunately, and let Paul go. Any pressing question out there? Okay, let's thank Paul for a great start to our workshop. All right. I think we're off to a great start. Paul set up the next couple of talks very well in his remarks about how green chemistry is already in many ways alive and well in the business world, and we're going to be hearing from two practitioners, if you will. I'm very excited to introduce both of them this morning. There was some thought given into how to structure this part of the agenda, and we wanted to really look at how green chemistry is percolating into the business models of a couple of companies, selected carefully here because we have a company that is primarily a chemical manufacturer but also has a foot in the downstream product arena. And we have a second company that's primarily a product manufacturer but also makes chemicals and understands that linkage in the supply chain. So let's start with, and I'll introduce both of our next speakers so we can keep things moving along. Mark Thompson is our first speaker in this next session. Mark is from the DuPont Corporation. He is the director of DuPont's Haskell Labs, Haskell Global Centers for Health and Environmental Sciences. I actually didn't know that whole name before, but and he in that capacity he oversees a lot of DuPont's work to really develop new products and chemicals. He came through the crop protection part of the company and led its discovery research division for a number of years before taking his current position. He actually joined DuPont in 1981, a freshly minted PhD in organic chemistry from Yale. And Mark is going to give us a perspective of a company at the upper end of the supply chain. Helen Holder is our second speaker in this session, and I've had the pleasure of working with Helen in a number of capacities. She is a corporate material selection manager. I think that's a very interesting, think about that, corporate material selection manager for Hewlett-Packard Company. And that is literally what she does. She spends much of her time painstakingly collecting information on the materials that go into her company's products, evaluating them, looking for every source of information she can in making wise selections that balance the kinds of factors that Dr. Anastas laid out earlier. Helen has also been very much of a player in the leading corporate efforts to try to advance both private sector and public policies that seek to advance some of the goals of green chemistry and sustainability. So it's my pleasure to start with Mark, and then we'll move to Helen.
I have to get set up with this thing also, so I can wander around. Is it on? Testing? Which side? Is it on? Can you hear me? Nope. Is there an on switch? Should
should be on. I
Okay.
think maybe just
All right. A little higher. How's that? That good? Okay. And this is advanced. Yep. Good. Okay. Thank you very much. Well, it's a real pleasure to be here today to talk about some of the things that DuPont does. I'm going to focus on R&D. And in fact, I'm going to concentrate really on the R part of R&D, on research. That's where I've spent most of my career. Paul and Richard did a great job of setting this up. Pardon me? Oh, it's off. All right. Fortunately, I think my voice carries pretty well. But all right, much better. Do I have to repeat? Reference was made to the pharmaceutical industry, and certainly great strides have been made, and it's really an honor to share the stage with many of the authors of a tremendous amount of literature describing new tools and approaches and methods. DuPont no longer has a pharma business, but we do have an ag business, which has tended to follow the pharma industry in science and technology about 5 years or so behind, developing many of the same tools, also looking for small biologically active molecules just for a different market space. And then the traditional chemical industry, the polymer chemistry industry, is moving up that learning curve very quickly. So just to put things in context for those of you who don't know about the current DuPont, this is how the R&D money was distributed last year, about $1.7 billion of investment. You can see that more than half of that was devoted to one of our megatrends, which is to feed the world. About 61%, this comprises our Pioneer hybrid seeds business, as well as the crop protection business, as well as nutrition and health. And then the rest of the R&D investment, which is still substantial, is distributed across other areas, including two other what we call megatrends, which is to reduce dependency on fossil fuels and also to protect people and the environment. That platform was really built on our Kevlar business, Nomex, and sustainable solutions. And of course, you expect some return on investment there for $1.7 billion. And what did it look like in 2010? DuPont introduced over the prior 3 to 4 years over $9.5 billion worth of new products. Last year alone, we introduced almost 1,800 new products. Now, these are not new chemical entities. These are not molecules that are new to the world. Some of them are, but they could be new pigments, new colors, new tests for food safety and that sort of thing, new fibers, as well as new molecules. So it's a very complex company, very difficult to get your arms around everything, but we do a good job, I believe. And one of the other messages from Paul was this stuff does work. There are good examples, which I'm going to share with you, mostly in the context of the ag business. You can see that we have a strong R&D presence around the world, and it is growing. From Meyrin, Switzerland, where we're doing photovoltaics and other research, to Shanghai in China. We established an R&D center a few years ago, and then just a couple of years ago, also an R&D center in Hyderabad, India. Expanding also in Brazil and putting things in such as next-generation biofuels, advanced protective materials, in addition to agricultural products. Now, to support all of this, DuPont nurtures a fairly complex network of technical competencies. I'm not sure how legible this is in the back, but it really comprises chemical sciences, biological sciences, material sciences, and then what we call leveraged technologies. And Haskell Global Centers is one of those leveraged technologies. Along the bottom, you see some others, such as engineering, analytical science, very key platforms, technology platforms that go across all businesses within DuPont. I'm honored to have been with Haskell Global Centers for about the past year as the director. And we focus on human toxicology, ecotoxicology, environmental sciences, which is really environmental fate and microbiology, as well as risk assessment and modeling. And when I talk about toxicology today, I'll be talking about all of those, not just human tox. So this is just a brief snapshot of Haskell Labs or Haskell Global Centers. We changed the name when we incorporated the Environmental Sciences Group into Haskell Lab about 3 years ago. And you can see under competencies some of the things we do, very traditional tox testing, environmental fate testing. And of course, we are expanding into endocrine disruption, more molecular toxicology, molecular biology, in silico profiling. We've done quite a bit in terms of computational toxicology, and I'll refer to some of that in a few minutes. We just celebrated our 75th birthday last year. Haskell was formed in 1935 in response to some concerns about bladder tumors in dye workers at DuPont, and we've enjoyed a lot of support since then. Now this slide really tries to describe what we mean when we think about green chemistry. I believe this quote was attributed to John Warner, that green chemistry is the molecular science of sustainability. If that was a misattribution, I apologize, but that's, I believe, who said that. And I think that hits the nail right on the head. DuPont has a strong sustainability program. It's really part of our fabric. It's embedded in our innovation process, and it's part of the way we work and we do business. Green chemistry and the principles associated with those, and as well as all the work that's been done over the past 10, 15 years or more, go into sustainability, supporting it, as well as green engineering. Just a couple of key points on this slide. The third bullet point there, implementing green chemistry principles. It's very knowledge intensive, pretty sophisticated science, and getting more so as we get down into the molecular level, down into the receptors, and not just focusing on whole organisms. As we develop sophisticated tools and methods to better understand mechanisms. And then finally, it's just a lot of hard work. We had a new scientist start with DuPont not long ago and said, you know, what's the secret? How do you get all this work done? And I just said, you just have to hang in there and do it. It's just a lot of hard work. So I think it was maybe Chris encouraged us to dream today, and this is a dream that we've had in R&D for many years, at least since computational chemistry came, and that is that a computational chemist could sit down at a computer screen, dial in all of the desired performance properties, physical chemical properties, tox, ecotox, environmental fate, as well as everything required to get that product to the marketplace. And the computer would spit out a molecule, maybe a handful of molecules, which the synthetic chemist could then go into the lab and synthesize. And oh, by the way, we also need a commercially feasible synthesis that's also green. No solvents would be preferred, but certainly green solvents, no waste, 100% yields, that sort of thing. If you're going to dream, you might as well dream wildly. Then just synthesize those handful of compounds, and one of those will be the product. You can scale it up, launch, get it registered. Now, we're not quite there yet, and under the category of hard work, just a lot of tools have been developed in various categories, starting with computational and silico tools. We found that just data mining, which is not terribly sexy, but just going in and being able to get to our data and then visualize it for the scientists really is a major step forward. In synthetic chemistry, things such as click chemistry from Barry Sharpless, Paul mentioned catalysis. Analytical chemistry is really the foundation upon which other sciences are built, especially chemistry. Performance efficacy testing, we've made a lot of strides there. High-throughput screening we've heard about. We tend to think of not only high-throughput screening, but appropriate throughput screening. You may not always need to screen 100,000 compounds a day. It may be 10 a week. It may be 20 in a month. But whatever that appropriate level would be is what we achieve. And then, of course, tremendous number of tools and methods and approaches under mammalian toxicology, from chemical characterization. Now you're starting to see some of the themes of toxicology in the 21st century come through. Tiered screening approaches, starting with in vitro assays, cell-based assays, and then moving to targeted in vivo screening. And DuPont also has a very strong animal welfare program, which is very important for us. Using surrogate species such as Daphnia is also part of our new assay development program. All of the omics technologies, proteomics, and then under environmental fate, with physical chemical properties Paul mentioned, modeling, and then persistence and bioaccumulation. And this would range from, say, a trout hepatocyte assay that we put in recently that gives us a very good bioconcentration factor number, to a landfill simulation, which we have set up in a lab, which is really an amazing operation to see. There's a guy who's studying garbology. So all of these things would fall under new tools and methods to get you answers quick, get you data quick back to the scientists. And then, of course, tox in the 21st century is something we're watching very closely. We're doing a lot of these things already. We are forming teams and collaborations to focus on human toxicity pathways, I think with a lot of other people in academia and other industries, other companies. Continuing to build on our expertise in computational modeling, computational toxicology. Again, I mentioned in vitro assays, and then targeted in vivo screening. And then the whole risk assessment process is undergoing a transformation, and we have 4 people who are involved in the ILSI-HESI-sponsored risk assessment in the 21st Century Initiative. We see a lot of benefits. Of course, I think the regulatory agencies see an opportunity to screen the 80,000 or so compounds in commerce in the U.S. very quickly and prioritize which of those should get further scrutiny. We see opportunities for getting more information earlier in the R&D process, doing it right the first time, minimizing waste, and it just makes good business sense. And we've seen examples of this. And then finally, accelerated commercialization times. I think for everyone doing research into chemistry, new molecules to the marketplace. And I've mentioned animal welfare, reduced animal testing would be a great ancillary benefit. Getting down to the molecular level is something we've done in our ag business, as has pharma, for a long time as we determine modes of action and sites of action for new classes of chemistry. So in the past, where we would just challenge the chemistry against whole organisms, we're now thinking about what happens at the gene level, what happens at the protein level, moving up to the cell, and then you gradually get into model systems, and then finally the whole organisms. So a lot of progress has been made in this area. Still a long way to go. One very key concept here, again, it falls under the category of not real sexy, but extremely important, is developing these assays in a way that gets data back to the research scientists very quickly, and does it in a way that's inexpensive, and then very importantly, doesn't require a lot of sample. So you're early on in a research program developing a new molecule, a new polymer type. You really can't be synthesizing hundreds and hundreds of grams of material for screens, for elaborate screens that may take months to run. I mentioned appropriate throughput. So if you need high throughput, you can do that. If you need 20 compounds every 2 weeks, you can also do that. So you tailor it to the desired throughput. And then finally, there's a good correlation with the real world. So if you've got an assay that's not really helping you move toward your desired target, it's really not doing you much good. I want to go back in time a little bit and talk about a rather old example, but it's still a good one, and it kind of lays the basis for some of the subsequent work that we did in the crop protection business. DuPont invented a class of herbicides in the late 70s called Sulfonylureas. It was a novel mode of action, inhibition of branched chain amino acid synthesis by inhibiting an enzyme only found in plants, so mammalian tox was almost a non-issue. Very favorable environmental profile. The use rates of these molecules took the industry from 1 or 2 kilograms per hectare, which is 2.5 acres, down to 50 grams or so, 50 to 100 grams. So a really very successful class of herbicides. There have been 4 other classes with the same mode of action developed. There are over 50 products that have been commercialized over the past 20 or 30 years, and they're still in use. 2 examples are shown at the bottom of products. But about a couple of years after the first product was introduced, we realized we had an issue with rotational crop safety. So for example, you control weeds in wheat. The following year, a farmer would plant another crop, a broadleaf crop, and that crop would be damaged by small amounts of the residual sulfonylurea herbicide because it was so potent. So we formed a multidisciplinary team. We had soil scientists, chemists, biologists, and they set up an assay, a lab-based soil degradation assay, and it had many of the characteristics that I mentioned. It mimicked the worst-case scenario in terms of soil type, pH, temperature. It was easy to run, inexpensive, didn't require a lot of sample. It took 2 weeks. The results came back to the scientists almost in real time. There were 5 categories of degradation, and it did a pretty good job of predicting, at least at the extremes. If you had something that was a Class A degrader, you knew it was probably only going to be too fast out in the real world. If it was at the other end in the lab, it was going to be too slow, and that was really a knockout. And you could eliminate a lot of dead-end molecules while they were still in the laboratory. And just a couple of learnings from this. There were many learnings, but a couple of learnings were that certain functional groups degrade in a pretty predictable way, such as nitriles, cyano groups, tend to hydrolyze, and then the amides tend to get chewed up by amidase enzymes. Those then degrade to carboxylic acids, which get bound up or degrade further. They at least become non-herbicidal. We also discovered some interesting molecular rearrangements, and one is shown at the bottom where the nitrogen atom actually comes back on itself and kicks out the whole middle section of the molecule, and it goes to something completely different, very innocuous, and that undergoes further degradation. But you take out that herbicidal activity. So it was a very successful program and has, I think, contributed to these molecules continuing to be very useful. Some sort of disciplined project management system is extremely important. This allows you to identify in which stage you need to do these laboratory assays, and you can continue to do this in a very disciplined and organized fashion. We've put in what we call a fail fast, and we also say succeed early to give it a little bit of a positive spin. It's consistent with a publication. Well, there have been a lot of publications about stage gate processes, but a few years ago in the Harvard Business Review, there was an article which was focused on pharma, which really said in the front part of research, you're looking to quickly eliminate poor candidates. In the second part, you're looking to successfully scale up. And then, of course, backup candidates are something that are very common in the pharmaceutical industry. What we found is that it's difficult to take a backup candidate too far along in your development process. It's carrying a couple of potential candidates, and you know you're going to drop one. This was put into place in our crop protection business in late 1999. Prior to this time, there was a lot of work that was done in the discovery phase, but it was not organized very well. And the crop protection business suffered a catastrophic collapse of the pipeline in 1999. And we knew we had to do something, so we put in more of a staged process and organized the testing so that in the first stage we were identifying those knockout factors so we could eliminate the bad projects early. We were also identifying issues that needed further investigation, and that was done in the next stage, which is really the heart of the discovery process, where you were solving things, you were getting around tox issues or environmental fate issues, and then finally in the last stage you were selecting among a handful of potential development candidates to go into full-fledged development. This has proved to be very successful. We've found that front-end loading in early research saves lots of time, lots of money, and lots of waste later in development. One of the first examples to come out of this process was a new insecticide called Rynaxypyr. This is just a picture of what it does. Plutella xylostella, or diamondback moth, really loves cabbage, and you can see that picture on the top. And with just a small amount of Rynaxypyr, about 10 grams per hectare, you have a pretty healthy-looking cabbage plant. It's been a successful product. It was launched a few years ago. One of the key things about this molecule is that it had a new mode of action, which is something that the insecticide business is always looking for. This compound inhibited the ryanidine receptor. And it was known for many years that this receptor controlled calcium flux and muscle contraction. And a natural product, ryanidine, was actually on the market, but it was toxic to mammals. It was taken off the market a few years ago. But Rynaxypyr was the first example of a synthetic insecticide that bound to this receptor. It was an agonist. It would open the channel, cause calcium to flow out irreversibly, causing paralysis and then death of the target insects. But the good news was, if you look at dose response curves for insects, and we've got roach, flies, and Heliothus, we're down in the nanomolar range in terms of inhibiting the receptor for Rynaxypyr. But if you test it against mammalian ryanidine, you're several orders of magnitude higher in concentration. So a tremendous safety factor there, which really gave us the confidence to go forward as quickly as possible. And then when it came to selecting the final compound, traditionally we would have picked the one with the highest efficacy. We picked the one with the best overall profile. It was not the most biologically active molecule in the final 6, but it had the best environmental profile and certainly the best tox profile. Nanomaterials is an area where DuPont has been active for the past decade, and I know, Richard, you've been very instrumental in putting together a nanorisk framework a few years ago, which provided a guideline, a 6-step process for identifying potential hazards and risks associated with nanomaterials. DuPont and the Environmental Defense Fund worked on this together. That has been complemented, I guess. I don't know if I can say superseded, but a new ISO standard has just been published in May of this year, which builds on this framework. There are some subtle changes. There are some more significant changes. I hear it's much more compact. And it also offers tiered tox testing approaches. So you start with in silico profiling, you move to in vitro assays that are validated, and then to targeted in vivo screening. DuPont has a very active product stewardship program and has just released an internal web-based process which basically allows you or requires now that you incorporate sustainability all the way from the beginning of your R&D process. And then at various milestone reviews and research reviews, those issues are brought up, questions are answered, resources are provided to help answer those questions. This slide just shows you that this process is well aligned with the 5 stages in the stage gate process. And not only are you focusing on the final product, but you can see words such as raw materials, supporting chemistry, intermediates, emissions, and waste are considered very early in the R&D process. You're not waiting till you're about ready to launch a new product. We've also developed a tool which looks a lot like ToxPy. This is a relational database that taps into 1,400 publicly available databases that allows you to assess hazard, potential risk, exposure, and even public perception. It's a very quick tool. It's being released internally. Our scientists are using this very heavily, and it gives you a very nice, easy-to-read pie graph. The red colors indicate potential issues in various categories around the pie. Yellow means maybe an issue there, maybe not, and then green means you're in good shape. So if you work your way around this, persistence, environmental persistence is the first category. Soil mobility. We've got bioaccumulation. Aquatic tox is considered. We're downward carcinogenicity, mutagenicity, reproductive toxicity. We move up to public perception. Are there issues around public perception with this class of chemistry? And then finally, environmental impacts, including transport and air. So this is it's a nice tool. We're keeping this. We've actually built on this every week. It gets better. And we'd be glad to share with you how it works and let you kick it around a little bit and maybe help us improve it. You can also rank chemistry. You can put in the CAS number or the structure or a substructure, put in a whole slew of compounds that are closely related, and it will spit out various pie graphs and show you at a quick glance which ones might be bad, which ones are probably better. It's not a decision-making tool, but it facilitates decision-making. And then let me just wrap up with we have an internal alternatives assessment framework. I think there are other alternative assessment frameworks out there. This is working well for us. It's tied into our Pro 3 program. And it's a 4-step process starting with identifying the chemistry at issue, why are you doing the assessment, and then you get into really a brainstorming session with a multidisciplinary team, looking at alternatives, including business feasibility. And this is the middle step. 2 and 3 are iterative. You're going back and forth until you find something which looks good. You fill out a template to compare various options, and then finally you decide among those alternatives. It's a very flexible system because we have such a diversity of products and businesses, and we recognize that one size does not fit all. This has just been implemented, and it'll get better with use. And then finally, I just want to kind of restate the themes here. Tremendous amount of progress has been made in developing tools. In silico, predictive tools are only going to get better. But in the meantime, we're going to implement the tox in the 21st century, risk assessment in the 21st century, lab-based tools, in vitro screens, cell-based assays. Some things are very important for doing R&D, multidisciplinary, well-integrated teams, a fail-fast, succeed-early approach that uses some sort of stage gate process. And then these highly effective knowledge feedback loops. Whatever your lab-based assay is, you meet those criteria: fast, inexpensive, low sample requirements, and validated. And with that, I want to thank you very much for your time and attention, and look forward to talking to you later.
Why don't we go directly to Helen and hold questions until after the 2 talks, okay? Since we're running just a bit behind.
You want this or do you want this?
Thank you, Mark.
Sure.
This is forward and back.
Thank you. Can you hear me? Are we on? Okay, great. So as Richard said, I am the Corporate Material Selection Manager at HP, and what we do is We write the specs and approve materials for use in our products. And that is a very interesting thing that we do right now. So today, I'd like to answer 4 questions. So why are electronics companies interested in green chemistry right now? What is green? Probably many of you have had to struggle with that question yourselves. How are we choosing better materials for our products? And what are we asking from you as chemists and toxicologists? So let's start from this beginning of why are we interested as an industry in green chemistry? In the late 90s, there were 2 laws that were passed in Europe that were very transformative for our industry in the material space, Ross and WE. These were 2 laws that were complementary, meant to function together, and dealt with the materials that were found in electronics. The purpose of them, the WE directive was intended to redirect material out of the waste stream. Electronic waste was a very big concern at that point. If you remember, the recycling of electronics waste was not actually particularly good at that point, and they were trying to redirect material. The companion law that went with this was ROS, Restrictions on the Use of Certain Hazardous Substances. And what it did was try to reduce the hazard, reduce the toxicity of the materials that were actually left in the waste stream. It restricted 6 materials, 6 substances: lead, mercury, cadmium, hexavalent chromium, and 2 classes of flame retardants, polybrominated diphenyl ethers and polybrominated biphenyls. This was a landmark law because it was the first reach into the material content of electronics to restrict what could be used. So we had already transitioned out of CFCs in the 80s in terms of our manufacturing processes, but this was really hit the core of our material set. By restricting lead, that really was the big one because we use tin lead solder. Solder is the most important material in any piece of electronics that you've got. It holds everything together. It makes it work. I love solder, and I will talk to you at length about it if you let me. But this was a huge deal. All of a sudden, we could no longer use this material that had been used for hundreds of years to connect metals. So this was, it turned out to be only the tip of the iceberg. Now we're starting to see a lot more materials restrictions in terms of the content of what can be in a product. So we're seeing, so ROS is actually written with a review cycle built in so that you have to consider new materials, new substances that might be added, and the elimination of existing exemptions. We're also seeing a lot more new jurisdictions. Our shorthand of this is China ROS or Korea ROS. They often are structured in a similar way and restrict the same substances. But more jurisdictions are getting into this game. And part of it is because they don't want to be left behind and also don't want to have product dumped there. So for example, in California, if you cannot sell a product in Europe because of the ROS restrictions, you cannot sell it in California either. They don't want to have an unequal requirement. And then also because of the REACH regulations in Europe, we're starting to see more reporting required. So when we started to do this implementation for July 1st, 2006, we used the traditional materials selection processes. So what did we look at? We looked at cost, function, manufacturability, reliability. And all unregulated substances were treated as equally acceptable. Now, there were a lot of people who were very concerned with this and in our industry. And there were kind of 2 reactions to this very legitimate concern about potentially making things worse with the replacements. So some parts of our supply chain decided that they were going to be obstructionist for all materials restrictions and say, okay, well, since we can never possibly know if something's going to be better or worse, we should not change anything ever. So that was one way. What we did was to say, well, let's figure out, can we evaluate these replacements to figure out if we can identify the better ones and make sure that we're not making things worse. Now, this was very much rooted in the fact that we have a grassroots pro-environmental movement inside of HP. But I'll be honest with you, that's not the only reason. As it happens, replacing substances is extremely expensive. There's a very compelling business reason to not commit yourself to a substance that you will then later have to remove with subsequent regulation. But think of it as your ATM fee, right? So every time you go to an out-of-network ATM, you're paying a fee. We would pay a fee. So if you changed from PBDEs to HBCDD as your flame retardant, oops, that's going to be restricted now. So now you're having to pay for twice that replacement. And if you're a bigger company like an HP, those ATM fees are in the tens of millions of dollars range. I don't want to pay a $10 million ATM fee. I don't know about you. But it's a very compelling business argument to make smart decisions about replacement technologies. Okay, so we decided we're going to do this. So now, what is green? Right? So this is a very tough question to answer, and we all deal with it every day, paper or plastic, I mean, we have to figure this out. So what did we do? We went out and looked at all the different tools, and here's just a sample. I'm not going to actually go through all these different tools, but if anyone would like to know why we did or did not adopt any of these, I'm happy to talk about it. But we did go out and we just tried a whole bunch of different things to see if we could come up with something that would let us make decisions. Actually, I'm going to just take a little detour for a second. We actually decide things. We actually approve materials, which means we're not advisors. We're not studying it for a bazillion years. We actually decide. In my inbox, I have questions. Can I use this? Should I use this or that? And I need an answer within 24 hours. Those are the kinds of things that we really have to deal with in the trenches when we're dealing with materials. So this was something, we couldn't have something that was going to take 12 years to do an assessment, and we couldn't do that. We needed something practical. And what a treat to have had Paul Anastas already make this argument for me, I have to say. Normally, when we start talking about doing this shift to hazard reduction, I'm usually in a pretty aggressively hostile environment of folks who are very much against using using hazard as a major screen. So I'm delighted to be in a friendly room and to have had him make this argument for me. Obviously, we all know this, right? Spills will happen, accidents will happen, exposures will happen. We want to get to a point where things are more benign already, and we just don't have to worry about exposure controls. We also have an interesting thing kind of in the electronics world that lets us make a simplification. I actually can't control exposure. If you would like to chew on our power cords, I can't stop you. If you would like to lick our circuit boards, I can't stop you. And I've heard they're delicious. I don't blame you, really. But for the same application, the exposures are the same. A power cord is a power cord is a power cord. And the brilliance of that is that it means we can actually take exposure out of a lot of these assessments. And we're able to, in the first pass, not completely, in the first pass, we're able to look at hazard as a primary consideration. It becomes a proxy. So what we do is we use comparative chemical hazard assessment. And what is this? This is where we're able to look at just really the hazard endpoints, carcinogenicity, persistence, bioaccumulation. We're able to narrow the scope down to focus on things that are measurable, things that are. So one of the difficulties that we have with LCAs is that you use a lot of proxies, you use a lot of estimations, and it's sometimes very far away from the data. By looking at hazard, we're able to narrow what we're looking at down and get a lot closer to things that are actually measurable. It's very science-based, and most importantly, we're able to screen out options. before we've invested a huge amount of money. It's the fail fast idea, right? So that we're deselecting options so we don't have to do a very big LCA. The tool that we use is the green screen for safer chemicals, which was developed by the NGO Clean Production Action. And I'm not going to go into a lot of detail on this. It's mostly, all this information is available on their website at cleanproduction.org. But I will just mention that it does cover 17 hazard topics that get scored on a high, medium, low scale with some thresholds. The thresholds are based on existing established thresholds. And it uses a decision logic to give an integer score from 1 to 4. So what I'm showing here is an example of how you might sort out the results on a particular substance. One of the things that we really like about this approach is that it gives us useful results. It's useful information for for decision making for specific products or specific cases. So you need to have an expert do this initially, because this is not for amateurs to go in and actually do this assessment. But once this is done, once you've got a score, these scores can now be used by non-toxicologists. Now this is really important because most materials that are selected, or that in all of these sort of engineered materials or engineered products that you might touch, doorknobs and chairs and whatever, all those materials are chosen by people who are not toxicologists. There's no vast conspiracy of people trying to put bad materials out in these products. We're choosing these materials based on function and all these other criteria. The number of board-certified toxicologists who are also procurement engineers is vanishingly small, right? So that's not what they do. They need to be able to make decisions, and this lets them do that, to differentiate between options. And interestingly, these categories of 1 through 4, when I first met Mark and Lauren and was looking at the green screen, I was determined to have a continuous score. This was, for some reason, very important to me. Maybe it just appealed to the scientist in me. I just wanted to have more resolution. As it turns out, this simple 1 through 4 is really powerful because now it makes these decisions easier. I can tell procurement people are really good with numbers. This is what they do. So I can say, no 1s. And they go, okay. It's done. And that decision is now made. Or it has to be better than what it's replacing, which is actually what we do. So if it's a 2 and you're replacing it, it's got to be better than a 2. If it's a 1 you're replacing, it's got to be better. This is actually really, really powerful. And very importantly, this meets our business needs. So this helps us to choose substances that are not going to be restricted in the future, because the regulating bodies are more and more using hazard as their screen. So this aligns our business process to what the regulators are doing. This also, very importantly, helps us to articulate goals to our suppliers. Everybody that we deal with believes that they have a very good sustainability program. Every supplier that we deal with absolutely thinks that they are doing everything they can for the environment. And to give them credit, they are often doing very, very good things. But this gives us a way to be specific about what we're asking, so that it's not bring me a rock. No, bring me another rock. No, I don't like that rock. Bring me another rock. Now we say, this is your goal. Your goal is to get better on this scale. And we have some cutoffs, but now they have their marching orders. And at the very beginning, we had some pushback. Again, sort of the hazard versus risk thing may be going on. But now we really have a lot of buy-in for this idea of trying to move up. So how are we choosing better materials? Again, we're screening on the green screen, but we still use all of the other, most of the other ways of doing materials decisions. So this is really busy. I'm not going to walk through all of it. But just to give you a sense, so once we identify a substance of concern, that's actually done for us. We just assume that we have to do that. We characterize its function, and we go out and look at our alternatives. This is where our hazard assessment is. We have given a prime place right at the beginning. right before you start doing even technical assessments, we're already deselecting anything that's not less hazardous than what it's replacing. Now, I've called this a parking lot because there is the possibility that going through the rest of the process, you may end up in a case where nothing actually works. So I've allowed for the possibility to go back. But in practice, that hasn't happened. And for any life cycle analysts out there, I would just like to apologize for the fact that LCA is in this lower corner. That means nothing in terms of its importance. It's just an artifact of the way the slide is set up. This really upset a few folks a while back. So after we do the hazard assessment and we find out if it'll meet our technical needs, then we do this life cycle thinking and look at exposure and do LCAs. And again, you don't want to have an LCA analyst spending time on something that's hazardous. One of the other things that you don't want to do is most LCA analysts will tell you that the toxicity portion of LCAs is actually not its strongest part. So one of the nice things, if you knock out the benchmark ones, when you pass the 2s, 3s, and 4s into the LCA, then they can actually do some of those trade-offs and not worry about making a poor decision on the toxicology. And that they really appreciate this because in general, LCA analysts are not toxicologists either. So this then makes a lot of sense. Oh, I can save a liter of water if I use a benchmark 2 instead of a 3. That is something you can live with. You don't want to say, I can save a liter of water if I use a PBT. Right? So we don't want to put them in a situation where they have to do that. This is just very briefly to say that we, my program actually engages far up with the formulators. So we don't deal with this sort of in-between in the supply chain. It's too complicated, and we just go straight to the formulators. But this is not just theory. This is fact. We do this. Our big program is our PVC-free power cords. We screen all of those materials. Mandatory. It's a mandatory screening program. It is requires full material disclosure. We've looked at about 30 materials, and several have been approved. The most important thing is that 100% of those PVC-free power cord formulations have been screened with the green screen. So every single one of those cords, all those materials have been screened. So what are we doing? We're bringing in all these different other types of materials. I mentioned that our group, we don't just do power cores. We actually look at solders and fluxes and printed circuit boards and surface finishes and all these different things. And we'll be bringing in those materials as we can. We have definitely been influencing our formulator culture. Again, when in the beginning, we got quite a bit of pushback, especially from the big companies. Interestingly, the mid-tier companies, they smelled the money. They know there's money here. And all of a sudden, they had access to a much, much bigger market than they had before. They got on board really quickly. And now the big companies have come around. So it's actually pretty cool. And again, we're encouraging the rest of our industry to use the same tools so that we can share information. So what do we need from you? The all-important question. What do we need from you? New and better materials. New and better materials. There are just not enough out there. There are not as many options as we need. Now, I have the benefit of working in an industry where we change our materials. We change our fundamental technologies, underlying technologies every day. So we're open. This is great, right? So we are flexible. We don't have years and years and years of legacy that we have to worry about necessarily. We want new materials. We want to design these features in from the beginning. Again, a theme you've probably heard already today. We want this from the beginning. We trained our formulators. We had a formulator class. And gosh, I think it was in February. After we taught them the green screen and we taught them what we wanted, someone raised their hand and said, I think we should probably screen these things before we get in and get them into formulation. To which I, of course, said, that's brilliant. I wish I had thought of that. No, it is a real revelation to some of these folks that you should really be doing this very far in advance of formulation. My promise to you is that I will keep the door open. So one of the downsides of being at a big company is that sometimes it's very easy to say no to new things, and especially because of the huge volume of material we need It's sometimes easier to say no. What I try to do is make sure that we can say yes. If you bring something new, if you've got a new idea, we're going to say yes. What else do we need? We need what you do. We need what you're doing here. We need fast, predictive ways of assessing the hazard of substances. Because we do have new materials. That may not be a 50-year-old post-World War II petrochemical-based substance. And I need an answer quickly. We really do use your work. And I want to say thank you for doing what you do. We really use it in our decision making. This is not theory. This is real. And my personal request is whatever you can do to make sure that chemists have toxicology training, please do it. This sounds like a really obvious thing, but it actually isn't. Most of the folks that we deal with in suppliers, they're very, very good chemists who have been doing their jobs for a very, very long time. And many of them have never, ever had a class in toxicology. They've never had to do anything, remotely related to toxicology. And so we find ourselves, me, the mechanical engineer in the room, educating chemists on toxicology. That's kind of crazy, right? We need to make sure that not just new graduates, but working professional chemists get to a point in their understanding that they're thinking about this as they're doing their formulations, as they're doing their synthesis today. So this thank you slide is not just a thank you for having me here. It is to just reiterate, thank you for the work that you do. As a far downstream user of it, it is really important to us that you do this work, and that you continue to do it. My vision for the electronics industry is that we get to a point where we no longer have regulation, echoing Chris's words this morning, that we don't have any regulations about what's in our products because the materials are so benign that no one cares anymore. So thank you very much.
Ask Mark to maybe come back up. We do have a few minutes for questions and comments, and I'd suggest we've heard two very intriguing presentations, different approaches being used by leading companies. So I would like to ask anybody in the audience who's got a question, either for one or both of the speakers, to start us off. Marty, you want to? I saw your hand first there, and Mark, why don't you come over to this microphone if you want?
Marty Stevens with the Humane Society of the United States. A question for Helen. So you mentioned a reliance on this green screen, which presumably uses legacy data to get a handle.
Yeah. Sorry, Marty. We're recording, so if folks could step to those mics, that'd be great.
Is this on? It's on. Okay. Martin Stevens with the Humane Society of the United States. A question for Helen. You mentioned reliance on the green screen as well as a need for new rapid tests. So presumably the green screen has a lot of sort of legacy data in it, and you look at new chemicals and their similarity to legacy chemicals and make some predictions based on the green screen. But when do you use these new rapid tests to give you the answers that you need, in some cases within 24 hours?
Well, the rapid, can you hear me? Okay. The rapid screening would be feeding into the individual endpoint scoring. So, for example, the endpoints would be your CMR, PBT, whatever. Those are where you would normally you'd like to have data. So we sometimes have to do analogs for different things. But if we had access to any sort of advanced assessment of new chemicals, that really helps us, so that we don't have to necessarily rely strictly on data that's not for a particular substance. So it's very deeply embedded, I guess, is what I'm saying. It's not at the logic level. It's very much in the actually scoring an endpoint.
So these new tests would be to amplify what you see in the green screen?
Right. So one of the things we do struggle with is a lack of data. Obviously, there are huge data gaps for lots of different substances. And anything that we can do to bridge the data gap, that's a good thing.
Pete Myers from Environmental Health Sciences. Thanks both of you for really, really informative presentations. It's impressive to see what both these companies are doing and how they're really trying to build a sustainable future that's consistent with the vision that Paul laid out. I've got questions for each of you, and I'll just ask both of them and then sit back. Mark, recently DuPont had to withdraw an herbicide that was really good on grass, but turned out inadvertently to kill trees. Have you stepped back and asked what in our screening process, what in our evaluation process failed? Was that a single event or a systemic problem? And how are you going about redressing it? And with respect to DuPont's work and the green screen, if you remember Paul's triangle with receptor-mediated processes up at the top and his acknowledgement that those were both potentially extremely important but also the least understood, how are you in the green screen prepared to incorporate that new science as it emerges? Because it's coming out really rapidly now, providing you with new tools and insights.
Okay, sure. I've been out of the crop protection business for about 3 years, but I'll do my best to give you my perspective, which will be somewhat limited. But I can assure you that there is an ongoing and in-depth investigation as to what happened, what are the causes, and DuPont very quickly responded and removed the product from at least those markets where they were seeing the injury. This was more of a selectivity issue between evergreens and weeds. So two types of plants basically, and one was non-target and the other was target. So you have lots of things going on, movement of the compound and the conditions and the temperature and the environment and that sort of thing. So I don't know the details, but given our sustainability program, the rapid response, and to work with the EPA and take the right measures quickly, and try to stop the problem as quickly as possible. And then, as I said, I can assure you there has already been a very in-depth investigation. I'm sure it's ongoing, and looking back to bringing the molecule forward, where could more testing have been done, if at all. And this is what happened after the catastrophic collapse of the pipeline in 1999. Several of those candidates were very advanced into development and suffered some pretty significant tox issues. And we went back and spent months looking back at what had been done and what could have been done better, and could we have identified the issue sooner. So just from a process standpoint, I'm sure that's going on. But as to the details of what happened, I can't say really. Helen?
Okay, so I actually do have a couple of backup slides that might help here. I can never go in without an eye chart somewhere in there. So in terms of integrating some of this work, one of the things that was really pretty cool about this, by the way, this framework is very much co-developed with EPA, DFE. So those folks are going to recognize all of this. One of the things that was really good about it is that there are placeholders in here, in particular for endocrine disruption. So at the time that they were creating this framework, there was a lot of concern, but there's really not a lot of data. There's not a lot of, at the time, there were not a lot of methods at all of doing anything with this endpoint. But they left it in here as a placeholder. And as your work develops, as you get better at this, these NDs mean no data. Why? Not just because there's no data, there is no data, but part of it is because there's no methods either. So as the science gets better on endocrine disruption, as the assays get better, this starts to fill in. So it's very much integrated in to already be looking forward in the science. One of the next areas that are being developed is how to adapt this for nano. Obviously, that's a big deal for us.
Okay, I think we're going to only have time for maybe one more question, but I am going to remind folks that both Helen and Mark are coming back. They're going to be back up here in panel discussions later in the meeting. So, and there's a break coming up. Please feel free to buttonhole them, but I'm going to have to ask Lynn to have the last question here.
Lynn Katz-Cherry from the Great Lakes Green Chemistry Network. I have two questions, too, and if there's not enough time, then we can do it later. But for Mark, I'm really interested in your slides to note that while I'm very impressed by all the work that's being done, it also seemed that in the end you were relying nonetheless on a risk model at the end of the process to determine what chemicals would be used. And so I'm just curious as to how you reconcile that with the emphasis that you've made on hazard early on, which, in some ways is not a good fit. And for Helen, I was just wondering if you addressed a little bit about endocrine disruptors, but how do you deal with areas where there's no data?
So yeah, ultimately, before we commercialize a new product, it does come down to a risk assessment, which would involve all of the information that we've collected over however long, months, if not years, on hazard. At times, an in-depth understanding of the potential routes of exposure, ingestion or dermal, or and coming up with a risk assessment. So I'm not. If I misunderstood your question, maybe we could talk at the break. I'm not sure I quite get it.
To design the hazard out, so you don't have to do a risk assessment.
Right. So yeah, there's certainly a prioritization process. So significant hazards, and there are thresholds, and they're very well defined for each of the stages. A hazard in a certain area will be a knockout, and that, for example, that first stage gate, no question about it. And some hazards can be designed around, and some cannot. And so those things that are not knocked out, we might identify issues and then get into that second stage. And if you can't get around them, that's when you have to kill it in the second stage. The attrition rate's pretty significant through those stage gates early on. Okay? All right.
So data gaps. Now, this is something that every working toxicologist struggles with all the time. So we face the same difficulties. We do have a minimum data set that we will normally ask for. What I will say, though, is that endocrine disruption, except in a handful of cases, is not always in that minimum data set, just because if we disqualified materials because of not having data for endocrine disruption, we would probably disqualify almost everything, and that doesn't help us. So the point is to differentiate as best we can with the information that we've got. That being said, it depends on the application, it depends on the chemical class. We will definitely adjust the minimum data set. So to give you an example, there are some replacements for PFAS, the perfluorinated compounds that have been synthesized that we were missing. They were missing carcinogenicity data. And we said, come on, right? You can't do that. So in that case, that was a really clear case. But for for phthalate plasticizers, you have to have that in there. You have to have endocrine disruption in there. So I don't want to say that it's absolute what the minimum data set is. We will adjust it, but we have to live with data gaps as long as it's something that we don't anticipate as being characteristic of that class. Right?
Okay, I'm afraid we're going to have to leave it there. We are going into a short break.